Hello, my name's Steve Hawkins. I'm the Product Marketing Manager from Meridian Bioscience. I will be presenting along with Sergi Leonardi from our R&D team, giving you some tips and considerations when developing your SARS-CoV/respiratory MDX assays. Before we start, I want to give you a very brief introduction to Meridian Bioscience. We are a multinational company that's been around for over 43 years now. Our focus is firstly on diagnostics in the areas of gastrointestinal, neonatal, pediatric and respiratory conditions, and secondly on life science with solutions for immunological assays and, more importantly with this talk, molecular assays. We are a company of innovation, being first to market with a C. diff test that proved that it was a hospital-acquired disease, and first to market with a H. pylori stool test. We were also first to bring a molecular point-of-care C. diff and also a malaria test to the market, and more recently developed the first LIO-ready RT-qPCR and air-dryable qPCR mixes. Our headquarters are in Cincinnati, but we also have locations across the US, as well as in Europe, with our molecular R&D team being based in the UK and our molecular manufacturing in Germany. We also have sites in the Middle East and in Asia. We are primary manufacturers of antibodies, antigens, and blockers, as well as molecular reagents, supplying not just the raw ingredients, but qPCR and RT-qPCR mixes that have been developed with the help and collaboration of our customers. We are now going to go into the tips and considerations, but let's first consider the process. Firstly, you set up your clinical question. For example, does this person have COVID? Or is this tissue cancerous, etc. You then take your clinical sample, release the nucleic acids from the cellular environment, and then isolate and purify it, and then go through a detection system to answer the question. This answer may be qualitative, a simple yes, no. Is this bacteria / virus there, or is the gene present, being expressed? Or it may be quantitative. How much is this gene being expressed? Or it may be that you're looking for a specific sequence, or you're looking for a mutation. Through this process, you need to take account of the challenges that you will face with cellular and environmental inhibitors, the sensitivity of the assay, assay design, what the format of the assay is, and, of course, the cost. And this is where I hand over to my colleague to go through these challenges and to talk about this.
Thanks for that, Steve. For this talk, I will be expanding on the aspects mentioned and talk through the challenges surrounding each subject. I will talk about sample inhibitors and factors such as patient variability, transport media, and sample preparation that can affect your reaction. I will then talk about sensitivity and achieving the limit of detection, and we will look into how pooling can affect this challenge. We will then look at reaction optimization to improve sensitivity. On top of the reaction chemistry, we will go through experimental steps to ensure that your results are reliable and free of potential false positives and false negatives. Finally, I will talk about considering format and how the customer will handle the product and what steps you can take as a developer to make things cost-effective. The first topic I would like to expand on is assay inhibition. Each type of sample will carry its unique set of molecular inhibitors that can impact your assay. This can be anywhere from the transport media, the extraction workflow, or more importantly, the sample in question. In this case, we are looking at sputum samples. These molecular inhibitors have multiple mechanisms at different levels. In the case of transport media, the reaction's chemical environment can be altered by the change in pH or ionic strength, while the dye is capable of absorbing your fluorescent signal. Inhibitors from a purification workflow, such as EDTA, can sequester the critical magnesium chloride in the reaction, or harsh chemicals such as SDS and phenol can denature the enzymes required for RT-qPCR. Finally, patient samples are complex systems and can have a range of mechanisms for inhibiting the reaction, either by interfering with the activity of the polymerase or by degrading key components such as target nucleic acids or enzymes. All these factors cumulatively lead to delayed CTs and low fluorescence signal as shown in the example here. So how do we overcome inhibition? One way is to consider the master mix. The formulation of the master mix can be crucial to tolerating the effects of the complex challenges of inhibition. As you can see here, we compare how a standard master mix compares to an inhibitor-tolerant one. Below, you can see that with increasing inhibitor concentration, the decrease in signal strength and delay in CT is much faster than in the standard master mix. However, even an inhibitor-tolerant mix will become encumbered, so it is important to adjust the sample concentration to avoid overloading the reaction with inhibitors. Overall, we recommend to use reagents that are compatible with your assay's inhibitory challenges. The next challenge I would like to address is sensitivity. Sensitivity in an RT-qPCR assay is demonstrated by earlier CT values and greater signal at low target input. The nature of the assay and target will define the limit of detection that is required to provide significant conclusions for your diagnostic test, and addressing sensitivity is important if your diagnostic test requires it. In this example, I compared two different master mixes in a template titration of 1000 to 10 copies per reaction. It is very clear that master mix A is more sensitive than master mix B, based on the definition we laid out. However, this could be a critical factor for a diagnostic test that aims to detect a target in low copy numbers. For example, if the limit of detection is 35 cycles for your assay to detect a particular pathogen, master mix A will give you a positive result, while master mix B will give you a false negative. One application that could be challenged by sensitivity is sample pooling. Pooling is a mathematical strategy that is used to eliminate groups of negative patient samples. In this particular schematic, you can see that rather than doing 27 tests, you're only doing 9 tests, thereby saving time and materials. Here, I've left a few references for guidance on how pooling can be implemented into making your workflow more efficient. While this method can dramatically improve a diagnostic workflow, this brings the challenges of decreasing the amount of target nucleic acid present by potentially diluting it with other negative samples. So, how do we address the challenges of sensitivity? Well, we've seen that sensitivity can be improved by using a more sensitive master mix, but what else can you do? The chance of detecting your target is completely dependent on the chemical and physical properties of your reaction. So let's break that down. The first point is to use a more concentrated master mix. This gives you the freedom to add more sample if required. Second are your primer and probes. The design and concentration of your primer and probes should be optimized on clean samples to ensure that you are getting the best efficiency out of your oligos. Another point is to optimize sample input by determining the minimum or maximum amounts of sample required to ensure reproducible results when amplifying positive samples. Finally, we highly recommend to follow the manufacturer's guidelines for thermal cycling conditions, as they can be specific per product. Also consider that if you are multiplexing, you may want to extend the time of your reverse transcription stage as well as your cDNA extension step. After addressing the chemistry of your reaction, you may want to consider some experimental optimizations. Two important challenges are false negatives and false positives. A false negative result is when you fail to detect the target that is present, and this can lead to a misdiagnosis and hence provide no action or treatment. In our particular situation, a patient with a false negative result for SARS-CoV-2 would then continue to transmit the virus rather than isolating. Several factors can contribute to a false negative, for example, an unsuccessful extraction, poor assay design, or a reaction that does not achieve the limit of detection. In order to address this, we would recommend to use an extraction control to rule out problems with your purification, use validated primer and probes if they are available, and follow the guidance we provided for improving sensitivity in the previous slide. The other two challenges I mentioned are format and cost. These two factors are very important when considering how you will get the assay to the customer. For example, do I need to cool my packaging? Will there be extra shipping costs for dry ice? Will my kit survive if delayed in customs? How can my customers store and handle the product to get the most out of it? The other two challenges I mentioned are format and cost. These two factors are very important when considering how you will get the assay to the customer. For example, do I need to cool my packaging? Will there be extra shipping costs for dry ice? Will my kit survive if delayed in customs? How can my customers store and handle the product to get the most out of it? So first I will talk about format. One tube or multi-tube. Here we refer to kits where components such as enzymes and buffers are pre-mixed or separated. Both formats have advantages and disadvantages with regards to ease of use, reproducibility, and freedom to optimize. The other important consideration I would like to introduce is the concept of wet versus dry. Dry mixes are those that are lyophilized or air-dried, and they can have advantages such as being able to add more sample, stable at ambient room temperature, and ease of use. You just have to add the sample. We recommend that you choose the format that will provide the stability and performance that your assay requires. I'd like to expand a little more on dried formats and compare lyophilized and air-dried. The two formats contrast in their appearance, cost, workflow, and applications. Lyophilization offers the freedom to dry down larger volumes, meanwhile air drying has a shorter drying time and can be done with standard equipment. Regardless of which dried format you select, they will both benefit from savings on shipping cost. With dried mixes, you are able to choose sustainable packaging and cheaper courier services without having to worry about losing activity due to delays in customs. All these benefits facilitate getting the assay to the customer, and more importantly now than ever in these pandemic times. Our last consideration is workflow efficiency. A shortage of purification kits has led to investigations into extraction-free workflows for detecting SARS-CoV-2. Such workflows involve a short sample pretreatment before direct addition of the sample to the reaction. These are documented in the references below. Implementing this workflow can make significant cost savings by saving time, cost of purification kits, and labor costs. We recommend this workflow using an inhibitor-tolerant mix that is able to perform in the presence of a pre-treated sample. So to finish the presentation, I would like to recap the challenges and solutions that we have talked about. When faced with inhibition, use a master mix that is optimized for the sample you are investigating. When tackling challenges of sensitivity, optimize the physical and chemical parameters of your reaction. When designing your assay, implement design and experimental controls to avoid false positives and false negatives. Finally, choose a format that delivers stability and performance to the end user, and consider the format that saves time and money. I hope you have found our tips helpful, and thank you for listening, and all the best with your development.